The influence of gut microbiota diversity on foraging intensity behaviors in Apis mellifera by Haley Eason. European honeybees, scientifically known as Apis mellifera, are very important parts of our ecosystem and pollination services in agriculture settings. They are the main pollinators of a huge percentage of the crops grown for food production and are critical to the production of roughly 30% of our global food supply crops. Honeybees are highly social, eusocial insects who have a division of labor among colony members, where differential tasks are assigned to various bees. Nurse bees tend to the hive, while forager bees collect nectar for carbs and pollen for protein. Foraging is important in honeybee colonies as foragers are responsible for gathering food and resources for the colony. The efficiency of honeybee colonies relies on task differentiation in the gut microbiota of honeybees may have an influence on this. The gut microbiota of Apis mellifera, mainly located in the honeybee's hindgut, is simple but has many technical roles. The gut microbiota plays a role in immune functions and host physiology, as well as aiding in detoxification, metabolism, and providing pathogen protection from various sources, such as pesticides. Since the gut microbiota houses bacteria that is essential to honeybee health and wellness, disrupting honeybee gut microbiota can cause a disruption in not only the health of the honeybee, but the honeybee colony as a whole, as they are eusocial species. The diversity of gut microbiome communities in honeybees is shaped by the host's environment. A recent evidence of the influence of gut microbiota on behaviors in honeybees suggests that microbes can impact the behaviors such as foraging. Understanding how the honeybee's gut microbiota can be influenced through microbes to produce a change in foraging behaviors is important for growing and maintaining bee populations and for healthy bee colonies. So my question is, how does the gut microbiota community of Apis mellifera influence foraging intensity behaviors through individual microbe inoculation interactions? A recent study was done that evaluated microbial diversity among nurse and forager bees and how the abundance of these specific microbes can affect their foraging behaviors. Scientists collected two groups of bees, foragers and nurse bees, and using a 16S rRNA gene amplicon sequencing test and a relative abundance analysis test, AMCON-BC, to determine relative abundance of each microbe in their nurse and forager honeybee samples. The purpose was to see which microbes were different in bees with different behavioral tasks. In the graph, the microbes that were of significant abundance difference are highlighted in yellow, and the three main ones of importance are B. asteroids, L. melventris, and B. melis. B. asteroids was of higher relative abundance in foragers, and L. melventris and B. melis was lower of abundance in foragers. The diversity of microbiota seen in foragers compared to nurse bees shows how variations in microbe abundance can play roles in the division of labor. The researchers then used this knowledge and went on to analyze the way that microbial communities of honeybees influences their foraging intensity. The scientists went on to test the influence of three individual microbes on honeybee foraging intensity behaviors. In 2021, they collected and treated groups with either B. asteroids, L. melventris, or B. melis, which was the treatment groups, or sterile food, which was the control. This allowed the researchers to have bees whose gut microbiota community composed of single honeybee-associated microbes or no honeybee-associated microbes. The bees were monitored using foraging analysis in an automated behavioral system called B-code to track the bees. Each bee had a custom matrix barcode that was unique for each individual bee, and their behavior was monitored for 6 days. Foraging intensity was determined by the proportion of foraging trips performed by the bees. At a group level, B. asteroid inoculated bees accounted for the majority of foraging trips during the first 2 days of being tracked. This can be seen in the top left graph, compared to the microbiota depleted bees. The top left graph shows the proportion of foraging trips per colony as a group, and the bottom represents the proportion of foraging trips per individual forager. At an individual level, day 1 of behavioral tracking was when individual B. asteroids inoculated foragers accounted for the majority of foraging trips among the colony. Forage intensity increased in the first 2 days after the inoculation compared to the microbiota depleted groups. This group also had a greater proportion of elite workers. Elite workers are honeybees that account for 50% or more of the foraging done by the colony. This can be seen in the right graph. In contrast to bee asteroids inoculated bees, L. melventris inoculated bees at the group level accounted for the minority of foraging trips performed the first 2 days of tracking, seen in the top left graph, compared to the microbiota depleted group. The individual foragers in this colony performed the minority of foraging trips also at the individual level, specifically on the second day of tracking, seen in the bottom left graph, compared to the microbiota depleted group. The group inoculated with L. melventris made up the minority of foraging trips at both the colony and individual level compared to the microbiota depleted group. Again seen in the left graphs. A difference in foraging intensity between B. mellis inoculated bees and the microbiota depleted bees was not seen at the group level, pictured in the top right graph. However, at the individual level, bees inoculated with B. mellis accounted for performance of the minority of foraging trips that took place during the first 2 days of tracking, as shown in the bottom right graph, compared to the microbiota depleted group. B. mellis had an influence on the foraging intensity at an individual level. Influencing the diversity in honeybee gut microbiota communities through individual microbe inoculation of specific microbes has an impact on the foraging intensity among honeybees. The relative abundance of bee asteroids seen in forager and gut microbiota communities is consistent with the increase in foraging intensity seen at both the individual and group level of bees inoculated with bee asteroids in comparison to the microbiota depleted group. The significant increase in foraging intensity seen in bee asteroids during the first 2 days of tracking shows that the microbe inoculated produced stronger effects the first couple of days prior to inoculation. Inoculating honeybees with either L. melventris or B. mellis microbes can decrease foraging intensity among colonies at the group level, but showed to only decrease foraging intensity at the individual level in the B. mellis inoculated group. These findings are consistent with the lower abundance of these two microbes seen in forager gut microbial communities. These findings are significant because they give insight on the influence of individual microbes on honeybee gut microbiota communities and their foraging intensity. Although not natural gut microbiome, inoculations of microbes can provide a path to influencing host phenotypes, specifically increasing forager intensity. A colony having efficient foragers is essential to the colony's health, and understanding the honeybee gut microbiota can help stop the decline in bee populations. Beekeepers could use inoculation of microbes as a way to influence foraging behaviors of their hive or other influential behaviors. These findings support the ability to modify the gut microbiome to alter behavioral trait variations in host phenotypes by inoculating with individual microbes that are specific to task specialization in bees. Thank you and have a good day.